Welcome back to the Deep Dive, where we take the world's most detailed knowledge, extract the crucial insights, and deliver them directly to you.
Today, we are diving into one of the most intellectually compelling paradoxes in all of molecular biology, the control of gene expression.
I mean, it's really the central challenge faced by every single living cell.
It is. And the paradox is, well, it's simple on the surface, but it's incredibly profound when you think about it.
Right. Every single cell in a multicellular organism, a tiny skin cell, a huge motor neuron, a liver cell, it contains the complete architectural blueprint.
The entire genome.
The entire thing. They all have the instructions for everything.
It's exactly like that analogy you had of a multi-purpose skyscraper blueprint. You've got the specs for the plumbing, the electrical grid, the ventilation.
And the rooftop garden, all in one giant book.
Exactly. But, if you're a 10th floor office worker, you only need the instructions for the coffee machine.
You definitely don't need the plans for the building's foundation right now.
Not at all. So our mission today is to understand how cells manage this information so precisely.
How they know which genes to express, which to silence, and when. And we're going to trace this system, following the logic of the source material exactly, from simple bacterial switches all the way to complex eukaryotic regulation.
And before we get into the nuts and bolts, it's worth just visualizing the sheer scale of this. The source material kicks off with this incredibly complex map of gene interactions in yeast. Saccharomyces cerevisiae.
And the numbers here are they're staggering. The researchers looked at something like 5.4 million combinations of mutant genes.
5.4 million. And what they were looking for is something called synthetic lethality.
Okay, what does that mean exactly?
It means, well, does mutating gene A and gene B at the same time give you a worse result than just mutating either one alone?
Ah, so it tells you if they're working together in the same pathway.
Precisely. It reveals a functional relationship. And the result of all that work was a map of 170,000 specific gene-gene interactions.
And that just tells you the most important thing right up front. No gene is an island.
Not at all. They're all nodes in a massive interconnected network. And we're about to unpack the rules that govern it.
So let's start with the source does, with the masters of efficiency. Prokaryotes.
Right. Their whole genome organization is just optimized for speed. The DNA is typically circular, double-stranded, and there's almost no wasted space.
Very little excess DNA, as they say. Almost everything codes for something.
And that structure helps with speed. On top of that, you see that genes that do the same job, say breaking down a certain sugar, are often clustered right next to each other.
Which allows for what they call coordinate regulation.
Exactly. So when the cell needs to do a task, all the tools, all the enzymes are made at the same time. And this brings us right to the core switching mechanism, the operon.
So let's use the two classic scenarios to explain this. Induction and repression. Imagine you move bacteria to a new dish.
Okay, so if you suddenly add lactose, the cell needs an enzyme, beta-galactosidase, to use it. Now, if the gene is off, the cell might have, say, 5 copies of that enzyme.
Just hanging around.
Right. But within minutes of adding lactose, that number shoots up by about 1,000-fold.
1,000-fold, wow.
Yeah, it's a massive, rapid switch on. That's induction. You can see it happen in real time. First, a spike in the mRNA, and then right after, the protein levels rise.
And the flip side is repression. So if the cell is making its own tryptophan and you suddenly give it a bunch,
it just slams on the brakes instantly. Yeah. The production of all the enzymes needed to make tryptophan is repressed, switched off.
And both of these responses are managed by the operon.
This brilliant system, figured out by Jacob and Monod, it really boils down to 4 interacting parts.
Okay, so you have the structural genes, the ones that actually code for the enzymes.
And they're all transcribed into one long, continuous mRNA molecule, a polycistronic mRNA.
Then you have the promoter, which is where the RNA polymerase binds to start transcription.
And right next to that, you have the critical part, the operator. This is the landing pad for the repressor protein.
And finally, there's the regulatory gene somewhere else that makes that repressor protein.
And the whole system, the whole switch, depends on the 3-dimensional shape of that repressor protein and whether its shape lets it grab onto the operator.
Let's look at the repressible TREP operon first. So if there's no tryptophan around,
the repressor protein that's made is naturally inactive. It just floats around. It can't bind to the operator.
So transcription just goes ahead. The cell makes the tryptophan it needs.
But when tryptophan levels get high, Tryptophan itself acts as a corepressor. It binds to that inactive repressor.
And changes its shape.
Completely changes its shape, activating it. This new active complex then clamps down on the operator and physically blocks RNA polymerase.
So the product of the pathway regulates its own production. It's a perfect feedback loop.
It is. Now, the inducible lac operon works the opposite way. By default, its repressor is naturally active.
So it's normally bound to the operator, blocking transcription. The system is off by default.
Which is great for saving energy, right? If there's no lactose, why make the enzymes to break it down?
Right, but when lactose shows up, it gets converted into something called allolactose.
And that is the true inducer.
Okay, so the inducer binds to the active repressor.
And again, it changes its shape. But this time, it changes it into an inactive form.
Causing it to fall off the operator.
Exactly. The roadblock is gone. RNA polymerase can now transcribe the genes, and the cell can use the lactose.
And as the lactose gets used up, the inducer disappears, and the repressor snaps back to its active shape and shuts the system down again.
This whole system of repression is what we call negative control, because the regulator, the repressor, inhibits the gene.
But the lac operon has another layer of control, a positive control system.
Yes, the famous catabolite repression, or as it's often called, the glucose effect.
This is where you see the cell's economic brain at work.
Yeah.
If you give E. coli both glucose and lactose,
it goes for the glucose every time. It's the better, more efficient fuel. It completely ignores the lactose until the glucose is all gone.
So how does it make that choice?
It's all about a signaling molecule, cyclic AMP or AKMP, and its level is inversely related to glucose.
So high glucose means low KMP.
And when glucose is scarce, CMP levels shoot up.
And that's the signal.
That's the green light. High levels of CAMP cause it to bind to another protein, the CAMP receptor protein, or CRP. This CAMP-CRP complex then binds to a specific site near the lac promoter.
And here's the really clever part of the design.
Yeah.
Even if lactose is present and the repressor is gone,
the lac promoter itself is actually pretty weak. RNA polymerase doesn't bind to it very efficiently on its own.
it's a low affinity site.
Exactly. But when the CAMP-CRP complex binds just upstream, it's like a molecular magnet. It physically bends the DNA in a way that makes that weak promoter suddenly a high-affinity, super-efficient site for RNA polymerase.
So you need two conditions met. Double check.
A perfect dual-gate system. The repressor has to be off, meaning lactose is present, and the CAMP-CRP complex has to be on, meaning glucose is low. Only then does the operon fire at full capacity.
Okay, beyond just starting transcription, the TREP operon has an even finer level of control called attenuation.
Yes, this is a second layer of feedback that actually controls transcription termination. It's incredibly sensitive to the amount of tryptophan.
So if tryptophan levels are high, transcription just stops dead in its tracks really early on.
Right, in a part of the mRNA called the leader sequence. But if tryptophan is low, transcription continues through all the genes.
And the mechanism is, it's just a masterpiece of RNA biology.
It really is. It links the speed of translation to the folding of the RNA. The leader sequence has 2 codons for tryptophan. If tryptophan is abundant, the ribosome zips right through that section.
And that fast movement causes the RNA upstream to fold into a very specific shape.
A hairpin loop that acts as a termination signal. It basically tells the RNA polymerase to stop and fall off.
But if tryptophan is scarce,
the ribosome stalls on those 2 codons, waiting for the right tRNA. And that pause, that traffic jam, allows the RNA to fold into a different shape.
an anti-terminator loop.
Exactly, a structure that lacks the termination signal, so the polymerase just keeps on going. The regulation is happening through the geometry of the RNA itself.
So finally, for bacteria, we have another class of regulators that feel almost ancient. Riboswitches.
They seem like a total throwback. They get rid of the regulatory proteins entirely.
The RNA regulates itself.
It does. A riboswitch is just a highly structured part of the mRNA, usually in the 5-foot untranslated region, that can directly bind to a small molecule, like an amino acid or a sugar.
And this is where the whole RNA world legacy idea comes from, isn't it?
It's a perfect example. The RNA itself is acting as the sensor. When the metabolite binds, the RNA molecule changes its shape, and that conformational shift physically blocks either transcription or translation. It's a protein's job, done by RNA alone.
Okay, so let's shift gears from the streamlined world of bacteria to the vast complexity of eukaryotes.
And right away, the control problem just explodes. You have two massive differences. First, the genome is huge, and it's locked away inside the nucleus.
Separating transcription from translation.
And second, the DNA isn't naked. It's packaged into this incredibly dense material called chromatin. It's a DNA protein complex.
So where bacterial regulators had a clear shot at the DNA, eukaryotic factors have to navigate this dense forest to find their targets.
And the packaging challenge itself is just mind-boggling. You're trying to fit about 10 centimeters of DNA from your largest chromosome into a nucleus that's only 10 micrometers across.
It's an insane level of compression that still has to be accessible.
And the solution to that puzzle is a class of small proteins called histones. They're full of positively charged amino acids like arginine and lysine,
Which lets them bind tightly to the negatively charged DNA backbone.
Right. And they're so fundamental that they're unbelievably conserved through evolution. The H4 histone in a pea plant and a cow differ by only 2 amino acids.
So the basic unit of this packaging is the nucleosome.
Correct. The core of it is 146 base pairs of DNA wrapped about 1.8 times around a central octamer of 8 histone proteins.
2 copies each of H2A, H2B, H3, and H4.
And this first level of packaging gets you a packing ratio of about 7:1. If you look at it under an electron microscope, it has that classic beads on a string look.
Right, assuming you've removed the linker histone, H1.
Yes, H1 sort of clamps the DNA down where it enters and exits the core, helping it coil up into the next level of structure.
But what's really fascinating about the nucleosome structure are the parts that stick out, the flexible N-terminal tails of the histones.
These tails are the real regulatory hub. They're not just structural, they project out, and they are covered in sites that can be chemically modified.
Acetylation, methylation, phosphorylation.
All of it. And this dynamic modification of the tails is what turns chromatin from simple packaging into an active regulatory surface.
So the next level of folding is the 30 nanometer fiber.
Which gets you to a packing ratio of about 40:1. The exact structure is still a bit debated, but the leading theory is the zigzag model.
Where alternating nucleosomes kind of stack on top of each other.
Exactly. And this coiling depends on that H1 linker histone and also on the interactions between those histone tails on neighboring nucleosomes. If you chop off the H4 tails, for instance, the fiber can't form properly.
And this fiber is then organized into even larger loops, anchored to a protein scaffold, all leading up to the most compact state we see.
The mitotic chromosome, which is packed down by a factor of 10,000:1.
So let's talk about the structure that holds all of this, the nuclear envelope.
It's a double membrane, and it's actually continuous with the rough ER. But the key feature is that these two membranes fuse together at thousands of points to form these openings.
And those openings are filled with the nuclear pore complex, or NPC.
Right, and just underneath the inner membrane, you have this protein meshwork called the nuclear lamina. It provides structural support.
And we know how important it is because of certain human diseases.
Absolutely. A mutation in the gene for lamin A causes a devastating disease called Hutchinson-Gilford progeria syndrome.
HGPS. It results in premature aging.
And it's because the misshapen lamin protein destabilizes the entire nuclear architecture. The nucleus itself gets deformed.
But the real action is the trafficking through that nuclear pore complex. The scale of this is hard to comprehend.
The numbers are wild. A single HeLa cell, a common lab cell line, has to import over 500,000 ribosomal proteins.
And export 14,000 ribosomal subunits.
Every single minute.
So it's not just an open hole in the membrane.
Oh, not at all. The NPC is a gigantic octagonal complex, far bigger than a ribosome, built from about 30 different proteins called nucleoporins.
And a T-type are the FG nucleoporins.
Yes, they have these flexible, disordered domains rich in phenylalanine glycine, or FG repeats, and these repeats fill the central channel of the pore,
forming a kind of hydrophobic mesh or sieve.
That's a great way to put it. It effectively blocks anything larger than about 40 kilodaltons from just diffusing through.
So how does all that massive cargo get across?
It needs a ticket, a molecular zip code called the nuclear localization signal, or NLS.
Which is usually a short stretch of positively charged amino acids.
Right. And in the cytoplasm, that NLS on the cargo protein gets recognized by a transport receptor called importin.
And that whole complex then docks at the pore and starts to move through.
It does, by making a series of very transient, quick interactions with those FG repeats, essentially hopping its way across the mesh.
But what drives it in one direction? What makes it go in and not back out?
That's a beautiful part. It's an energy gradient provided by a small GTP binding protein called Ran,
Okay, so how does that gradient work?
There's an enzyme in the nucleus that keeps Ran loaded with GTP, so you have a high concentration of Ran-GTP inside the nucleus.
And an enzyme in the cytoplasm does the opposite, so you have low Ran-GTP outside.
Exactly. The energy from GTP hydrolysis maintains that asymmetry. So when the important cargo complex finally gets into the nucleus,
It encounters that high concentration of Ran-GTP.
right, and Ran-GTP binds to the important, which forces it to release its cargo. The delivery is made.
And then the important, now bound to Ran-GTP, gets shuttled back out to the cytoplasm.
Where the GTP is hydrolyzed, Ran-GTP is released, and the important is ready for another round. And protein export just works in reverse. Ran-GTP promotes the assembly of the export complex in the nucleus.
And for RNA, it's all about quality control. Only fully processed, mature mRNAs get the export signal.
Right. So now let's look inside the nucleus at how chromatin itself is organized into different functional states. We're talking about euchromatin and heterochromatin.
Euchromatin is the one that's more dispersed, right? The transcriptionally active stuff.
Yes, it's the majority of the chromatin. Heterochromatin, on the other hand, is super compacted, it stains very darkly, and it's generally silent, transcriptionally inactive.
You usually find it clumped up near the edge of the nucleus by the lamina.
And we can split it into two types. First, there's constitutive heterochromatin. This is stuff that's permanently silenced. Think of centromeres and telomeres.
And its silencing is so powerful it can spread. It has this position effect.
It does. If you move a normally active gene next to it, it'll often get shut down.
The other type is facultative heterochromatin.
Which is more dynamic. It's chromatin that gets inactivated only in specific cells or at certain times. And the most famous example of this is X-chromosome inactivation.
In female mammals, right? To balance the gene dosage with males.
Exactly. Since females have two X chromosomes, one gets almost completely shut down and condensed into a structure called the bar body.
And which one gets inactivated, the one from the mother or the one from the father, is a random choice early in development.
Which leads to the amazing fact that all female mammals are genetic mosaics.
We can see this so clearly in calico cats. The patches of black and orange fur are just large clones of cells where either the X with the black allele or the X with the orange allele was randomly silenced.
And the process is initiated by a fascinating molecule called sexy2 RNA. It's a long non-coding RNA.
So it's an RNA that doesn't make a protein.
Right. It's transcribed from the X chromosome that's about to be silenced, and the RNA transcript then physically coats that entire chromosome, recording protein complexes that establish and maintain the silenced state.
So switching between these states, from active euchromin to silent heterochromatin, brings us right back to the histone code.
It all comes down to the histone tails. The idea is that the transcriptional state of a region is determined by the specific pattern of chemical modifications on those tails.
For example, repressive marks often involve the methylation of a specific residue, histone 3 lysine 9, or H3K9.
Whereas active chromatin tends to have acetylation at that very same spot. It's a code.
So let's walk through the process of forming heterochromatin. It's not just a one-off event, right? It's actively maintained.
It's a self-perpetuating cycle. So step one, a repressor protein will often recruit a histone deacetylase, or HDAC, to remove any activating acetyl groups.
Wiping the slate clean.
Step two, a specific histone methyltransferase is recruited, and it adds that repressive methyl group to H3K9.
And step 3 is the key to making it stick.
This is the critical step. That methylated H3K9 becomes a docking site, a landing pad, for a protein called heterochromatic protein 1, or HP1.
And HP1 doesn't just sit there.
No, it acts as a bridge. It binds to the methylated histone, but it also recruits more of the methyltransferase and interacts with other HP1 molecules on nearby nucleosomes.
Creating this feedback loop that spreads the silencing mark along the chromosome.
Exactly. It builds this interconnected, compacted network. That's the molecular basis for how heterochromatin spreads and silences genes.
And we should also mention that small RNAs from repetitive parts of the genome actually help guide this whole machinery to the right places to begin with.
Yes, they play a crucial role in targeting.
Okay, so if we zoom out to the fully condensed mitotic chromosome, this is what clinicians look at in a karyotype.
Right, where they arrange the homologous pairs by size to screen for large-scale chromosomal aberrations.
Like breaks, deletions, or translocations. An inversion, where a piece gets flipped around, can cause major problems during meiosis.
And a translocation where a piece of one chromosome gets stuck onto another can have huge consequences. The classic example is the Philadelphia chromosome,
Which causes chronic myelogenous leukemia, or CML.
It's a translocation between chromosome 9 and 22 that creates a new fusion protein, BCR-ABL$, which is a constitutively active kinase that drives the cancer. It's a direct link from a structural change to a disease.
And these translocations are also engines of evolution. The source points out that human chromosome 2 is actually the result of a fusion of two smaller ape chromosomes.
Yes, which is why our haploid number is 23, not 24. We also see deletions, which cause syndromes like Cray-Duchat, and duplications, which can mess up gene dosage.
Let's focus on the special structures at the ends of the chromosomes, the telomeres.
Since our chromosomes are linear, we need these protective caps. They're made of thousands of repeats of the sequence TTG.
And their main job is to solve the end replication problem.
Right. Standard DNA polymerase can't copy the very end of the lagging strand, so with every round of cell division, the chromosome would get a little bit shorter.
Until you start losing important genes.
But the solution is an amazing enzyme called telomerase. It's a reverse transcriptase that carries its own RNA template with it.
And it uses that template to add new TTG repeats onto the end of the chromosome, extending it.
Which then gives the regular DNA polymerase enough room to come in and fill the gap. It's essential for protecting the chromosome ends.
And telomeres are directly linked to aging and cancer. Most of our normal body cells, our somatic cells, don't have active telomerase.
So their telomeres shorten with every division, which eventually triggers replicative senescence. It's a built-in clock, the Hayflick limit.
But cancer cells find a way to cheat death.
About 90% of human tumors manage to reactivate telomerase, which gives them a form of immortality and allows for uncontrolled growth.
Okay, now to the middle, the centromere. This is the constricted part of the mitotic chromosome where the kinetochore assembles.
It's the attachment point for the microtubules that pull the chromosomes apart. And what's really strange about centromeres is that their identity isn't really defined by the DNA sequence.
So it's epigenetic.
It's a perfect example of epigenetics. The defining feature is a unique histone variant called CENPA, which replaces the normal H-cre histone only in the centromeric nucleosomes. That protein is the real mark, the platform for building the kinetochore.
Which brings us back to the formal definition of epigenetics. Heritable information that's not encoded in the DNA sequence itself.
Like X inactivation, like the histone code. And the key is that these states have to be passed down through cell division.
How does that memory get passed on during replication?
It's a template-based system. When the DNA is replicated, the old, modified histones are sort of randomly distributed between the two new daughter strands.
So each new strand gets a mix of old marked histones and new blank ones.
Exactly. And the enzymes that made the original marks, like the methyltransferases, travel along with the replication machinery. They recognize the old marks on the parental histones and copy them onto the adjacent new ones.
Perpetuating the code.
It perpetuates the chromatin state. This is how a liver cell makes more liver cells. And it's also why identical twins who have the same DNA can have different health outcomes. Their epigenomes can diverge over time.
So finally, we have to get rid of the idea that the nucleus is just a bag of chromatin. It's highly organized.
It is. During interphase, each chromosome occupies its own distinct space, a chromosome territory.
And their location isn't random. Gene-poor chromosomes tend to be out at the periphery, near the nuclear lamina.
While gene-rich chromosomes are more in the center, the inactive X chromosome is also pushed to the edge.
And genes on totally different chromosomes can come together to be regulated.
Yes, techniques like 3C have shown this. For example, after estrogen stimulation, two genes on two different chromosomes can physically move into close proximity.
Which supports the idea of transcription factories.
The idea that active genes move to specific locations in the nucleus where all the transcription machinery is concentrated. Even the splicing machinery is organized into these dynamic storage depots called speckles.
So, now that we have the lay of the land, let's look at the overview of eukaryotic gene regulation, starting with a really fundamental concept. Genomic equivalence.
Right. The big question for a long time was, when a cell differentiates, becomes a muscle cell, say, does it throw away all the genes it doesn't need?
And the answer, thanks to some incredible experiments, is a definitive no.
Stewart showed you could grow a whole carrot plant from a single differentiated root cell. Gurdon cloned a frog from the nucleus of a tadpole intestinal cell.
And of course, the most famous example, Dolly the sheep.
Cloned from the nucleus of an adult mammary gland cell.
So what all these experiments prove is that the specialized state of a cell is not permanent. The nucleus retains the entire genome.
And it can be completely reprogrammed by factors in the cytoplasm of an egg cell. The epigenetic marks can be wiped clean.
Which is good, because the regulatory challenge is just immense. A red blood cell has to dedicate over 95% of its protein synthesis to hemoglobin, which comes from genes that are less than 1/1,000,000th of its total DNA.
That kind of precision requires multiple layers of control. And the source breaks it down into four main levels.
First is transcriptional control.
Deciding if a gene is even transcribed and how often.
Second, processing control.
How that initial RNA transcript is spliced and modified into a mature mRNA.
Third, translational control.
Whether that mRNA in the cytoplasm actually gets translated into a protein.
And finally, post-translational control.
Regulating the activity and the lifespan of the protein itself after it's been made.
Let's dive into the first and probably most important level, transcriptional control.
And the game really changed here when technology allowed us to look at all the genes at once, not just one at a time.
We're talking about DNA microarrays and, more recently, RNA-seq.
Right. A microarray is basically a comparative experiment on a glass slide. You have thousands of tiny spots, and each spot contains the DNA for a specific gene.
And then you take two different cell populations, like yeast-fed glucose versus yeast-fed ethanol, and you label their mRNA with different colored fluorescent dyes.
Usually red and green. If you then mix those labeled probes together and wash them over the chip.
And the color of each spot tells you the story. If a spot is bright green, that gene was turned way up in the glucose-fed yeast.
If it's red, it was up in the ethanol-fed yeast. And if it's yellow, it was on in both. It gives you this beautiful, immediate visual snapshot of the cell's response.
But the more modern, more powerful technique is RNA-seq.
It is. Instead of relying on hybridization, you just directly sequence all the mRNA from the cell. It gives you an exact digital count of every transcript. It's incredibly sensitive.
And this has huge clinical implications for things like cancer treatment.
Absolutely. You can take a tumor, sequence its RNA, and get a precise gene expression signature. You can see exactly which genes are driving that specific cancer.
And that allows for personalized medicine. You can choose a drug that targets the specific pathway that's active in that patient's tumor.
It's moving away from one-size-fits-all treatment.
So the molecules that are orchestrating all of this are the transcription factors, or TFs.
And we can split them into two main groups. You have the general TFs that are part of the basic transcription machine at the promoter.
And then you have the sequence-specific TFs that bind to regulatory sites and act as either activators or repressors.
And almost all gene control is combinatorial. It's not one TF flipping a switch. It's the specific combination of multiple TFs binding to a gene's control region that determines the outcome.
These TFs are so powerful, they can literally define a cell's identity.
Right. The TF called MyoD is a master regulatory factor. If you force a connective tissue cell to express MyoD, it will turn into a muscle cell.
That's incredible.
And even more incredible is the work of Yamanaka, who showed you could erase a cell's identity entirely. He used just 4 specific TFs.
OCT4, SOX2, MyMic, and KLF4.
To reprogram adult skin cells back into a pluripotent embryonic-like state, creating induced pluripotent stem cells, or iPS cells.
Wiping their epigenetic memory clean.
So structurally, these TFs all have a DNA binding domain and an activation domain, and they very often work as dimers, which greatly expands their regulatory potential.
And they use a few common structural designs, or motifs, to bind DNA.
One of the most common is the zinc finger. A zinc ion helps fold a part of the protein into a finger-like shape that pokes into the major groove of the DNA.
Then there's the helix loop helix, or HLH motif,
which critically always functions as a dimer. And because they can form heterodimers, mix and match pairs, you can generate a huge amount of regulatory diversity from a small number of proteins.
And the third major one is the leucine zipper,
where a repeating pattern of leucine residues on an alpha helix causes 2 of them to zip together into a coiled coil, forming the dimer.
And all of these TFs have to find their specific landing sites on the DNA.
Which are the DNA regulatory sites. You have the core promoter with the TATA box that sets the start site. And then you have the proximal promoter region upstream with sites like the CAT box and GC box that control the frequency of transcription.
And a huge number of genes, maybe half of them, have alternative promoters.
Which means the same gene can be transcribed starting from different points, which can lead to different versions of the protein. It's another way to expand the proteome.
So experimentally, how do we find where these TFs are binding?
A powerful genome-wide method is called chromatin immunoprecipitation, or CHIP.
Okay, break that down for us.
First, you use a chemical, like formaldehyde, to cross-link the TFs to the DNA they're currently bound to. Then you shear the chromatin into small pieces.
And then you use an antibody that's specific for your TF of interest.
Exactly. The antibody grabs the TF, and because it's cross-linked, it pulls the DNA it was bound to down with it.
You immunoprecipitate the complex.
Right. Then you reverse the crosslinks, purify that DNA, and sequence it. That tells you every single place in the entire genome where that TF was bound.
And what this has shown is that many of these binding sites are really far away from the gene's promoter.
Which brings us to the concept of enhancers. These are DNA elements, often thousands of base pairs away, that contain clusters of TF binding sites.
And they can boost transcription by 100-fold or more. But how do they do that from so far away?
The intervening DNA forms a loop. It physically brings the enhancer with its bound activator proteins into close proximity with the core promoter.
And those activators don't touch the polymerase directly. They recruit intermediary complexes called coactivators.
Right. And these coactivators do two main things. Some of them, like the mediator complex, interact with the basal transcription machinery.
And the other group are chromatin remodelers.
They are absolutely essential because their job is to deal with the physical barrier of the nucleosomes. They modify the histone tails to open up the chromatin.
This is the histone code in action. The key activating enzymes are histone acetyltransferases, or HATs.
They add acetyl groups to lysines on the histone tails, which neutralizes their positive charge and loosens their grip on the DNA.
So let's trace the whole activation sequence. An activator TF binds to an enhancer.
It then recruits a coactivator that has HAT activity. That coactivator acetylates the nearby histones.
And that acetylation mark acts as a binding site for another set of machines.
The chromatin remodeling complexes, like SWES-NFF.
And these use the energy from ATP to physically shove the nucleosomes around.
They can do a few things. They can slide the nucleosome along the DNA to uncover the promoter. They can temporarily eject the nucleosome entirely, or they can swap out standard histones for variants that make the chromatin more open.
So it's a multi-step process of clearing the way for the transcription machinery to assemble.
Precisely.
Of course, there's also transcriptional repression.
Which often works by reversing that process. Repressor proteins recruit corepressor complexes that contain histone deacylases, or HDACs.
They remove the acetyl marks.
Which then allows for the addition of repressive methylation marks, like H3K9 methylation, leading to HP1 binding, and the formation of silent heterochromatin.
There's one more major repressive mechanism, DNA methylation.
This is the addition of a methyl group directly onto cytosine bases in the DNA, specifically at CPG sites.
And this methylation pattern is heritable. It's copied after DNA replication.
Yes. And heavily methylated promoters are almost always associated with silent genes. It's a very stable, long-term silencing mark.
This is also the basis for genomic imprinting.
A really bizarre phenomenon where a gene's activity depends on whether you inherited it from your mother or your father. The IGF2 gene, for example, is only active from the paternal copy.
And this is controlled by sex-specific DNA methylation patterns that are established in the sperm or egg.
Right. And if that pattern is disrupted, it can lead to diseases like Prader-Willi syndrome.
Finally, we have to mention long non-coding RNAs as repressors.
Yes. Like the Hotera LNC RNA we talked about, it acts as a scaffold. One end grabs one repressive complex, the other end grabs another, and it delivers them both to a specific target gene to shut it down.
Okay, so once a transcript is made, we're not done. We move to RNA processing control.
And the superstar here is alternative splicing.
This is how we get so much complexity from a relatively small number of genes.
It's the primary proteome expander. A single gene can produce many different proteins just by splicing the primary transcript in different ways.
Like the fibronectin gene, which makes one version for the cell matrix and another, shorter version for the blood plasma.
Or the mind-blowing example of the DSCAM gene in fruit flies. Through combinatorial alternative splicing of 4 different exon clusters.
It can produce over 38,000 different proteins from a single gene.
38,106 to be exact. It just completely demolishes the old one gene, one protein idea.
And this splicing choice is regulated by proteins that bind to the RNA, either promoting or inhibiting the use of certain splice sites.
There's also RNA editing, where the sequence of the RNA is chemically changed after it's been transcribed.
Like changing an adenosine to an inosine, which the ribosome reads as a guanine.
Or the textbook example of the apolipoprotein B gene. In the intestine, a specific cytidine is edited to a uridine,
which creates a premature stop codon.
Right. So the intestine makes a short, truncated version of the protein called ApoB48, while the liver makes the full-length version B100, same gene, different proteins because of one little edit.
All right, last major checkpoint, translational control in the cytoplasm.
This is often controlled by sequences in the untranslated regions, or UTRs, at the ends of the mRNA.
A great example of translational activation is what happens in a frog egg after fertilization.
Before fertilization, there are all these maternal mRNAs just stored in the egg. Translationally silent, they're held in check by a protein called maskin.
But after fertilization, a signal causes maskin to be removed, and an enzyme rapidly adds a long poly-A tail to these mRNAs.
And that long tail is the signal to the ribosome. Translate me now. And you get this huge burst of protein synthesis needed for early development.
The cell can also shut down translation on a global scale during stress.
Yes, a number of stress pathways all converge on phosphorylating one key initiation factor, EIF2. When it's phosphorylated, it shuts down almost all protein synthesis in the cell.
But control can also be very specific, like the mRNA for ferritin, the iron storage protein.
Its 5' UTR contains a sequence called the iron response element, or IRE. When iron levels are low, a protein called IRP binds to the IRE.
And physically blocks the ribosome from translating it. No need to make a storage protein if there's no iron to store.
But when iron is high, iron binds to the IRP, it falls off the mRNA, and translation begins. It's a simple, elegant sensor.
And another layer of control is the cytoplasmic localization of mRNAs.
Getting the message to the right place in the cell before it's translated. In a fly embryo, for example, the bicoid mRNA is shipped to the anterior end to define the head, while Oscar mRNA goes to the posterior to define the germ cells.
Finally, there's the stability of the message itself, its mRNA half-life.
How long an mRNA sticks around determines how much protein can be made from it. Some mRNAs, for proteins needed in quick bursts, have destabilizing sequences in their 3' UTR.
And these sequences are recognized by proteins and microRNAs that trigger the rapid destruction of the mRNA.
If you delete that sequence from a gene like FOS, its mRNA becomes hyperstable, which can be a direct cause of cancer.
What a journey. From bacterial switches all the way to the stability of the message in the cytoplasm.
It really shows you that gene expression isn't one single event. It's a whole pipeline of control points, a series of checkpoints from start to finish.
We started with the rapid on-off logic of bacterial operons, and then moved into the slow, architectural, chromatin-based control in eukaryotes.
Where the histone code and DNA methylation act as this fundamental memory system defining the landscape that the transcription factors have to navigate.
And yet within that stable system, you have this incredible flexibility from mechanisms like alternative splicing, which lets a gene like DSCAM create a proteome's worth of diversity all on its own.
I think if there's one central theme, it's this constant tension between stability and plasticity. Our identity as a cell type is rooted in the stable, heritable maintenance of those epigenetic marks.
But then you have systems like genomic imprinting, which show that this entire system has to be erased and rewritten every single generation.
It's that duality that allows for the complexity of development, but it also creates vulnerabilities that can lead to disease. It's just a truly elegant integration of structure, energy, and information.
Thank you for joining us on this deep dive into the control of gene expression. We look forward to exploring the next frontier of molecular biology with you soon.